Hi, good afternoon. My name is Beatriz Adriana Valverde Fox, and today I'm going to present the article Learn them the rice, crayfish-turtle, co-culture maintains high crop yields by improving soil health and increasing soil microbial community stability. Okay, the introduction. A monoculture farming has led to agricultural intensification in recent years. Despite the high yields generated by these practices, monoculture has led to considerable negative impacts on environment, ecosystems, including decreased biodiversity, excessive of the synthetic pesticides, and mineral fertilizers, and deterioration of soil health. Farming systems diversification via the culture of crops and aquatic animals has been considered an effective means to develop ecological agriculture while increasing agricultural biodiversity and ecologic sustainability. Rice, crayfish, and turtle co-cultures can be used to establish new agricultural systems with more complex species combination to ultimately increase income and ecological health. And these practices can aid in the efficient use of available energy and nutrients and have been widely recognized as a way to sustainable manage agricultural system. Okay. The sustainability of agricultural management practices can be assessed by measuring soil health indicators over time. Soil microbial diversity and community structure can effectively reflect soil health across management practices and thus can indicate crop productivity and be used as indicator of predictions of soil health. Moreover, soil microbial communities play critical roles in ecosystem stability and sustainability. Microbial community assembly is the primary mechanism underlying microbial interaction and is regulated by combined deterministic and stochastic processes. And previous studies has demonstrated that rise in aquatic animals farming systems can increase agricultural biodiversity and rice production by promoting beneficial biotic interaction and improving soil fertility quality and also affect soil microbial community structure. This study aimed to understand how co-occurrence network and soil microbial community stability respond to long-term RCT management, how the relative influences of assembly processes regulate soil microbial community change, and RCTs when formed for different years, and what co-association partners exist among soil health and microbiome stability as they relate the medianly rice yields under RCT culture system. Okay, material and methods in the experimental area and design. Okay, this study conducted at a large farm affiliated with Shanghai. Shanron Aquaculture Professional Cooperative located in Dadong Village in Shuxing town within the Xiangmin district of Shanghai, China. Um, in this part of the experiment, rice seedling was transplanted from a nursery into Yale's plots every May. Young crayfish, 5 grams each, and turtles, 100 grams each, were released into the field plots and a standard abundance of 45,000 and 900 juveniles per hectare respectively, and the crayfish and turtles were then allowed to self-propagate inside the rice paddies, and only basal fertilizer was used for rice cultivation. Okay, the next part of the experiment is soil sampling and storage. First, soil samples were collected from age of the 12 years RCT, a 7-year RCT, and RM treatment plots and depths of 0 and 30 centimeters using a sample probe during the rice mature stage on September 25, 2019. Next, a total of 45 soil samples were collected and were placed on ice and immediately transported to the lab. And finally, after passing through a 2 millimeters sieve, a portion of the soil sample was stored at a 80 Celsius degree prior to DNA extraction, while the remainder was stored at 4 Celsius degree prior, prior to analysis of soil properties. In the soil physico-chemical properties and microbial community DNA extraction, first the pH was determined by preparing a suspension of an air-dried soil sample, and measurement was carried out with a digital pH meter. Next, soil organic carbon, total nitrogen, total phosphorus, and total potassium were determined. Next, the result samples were analyzed using molybdenum blue flame spectrophotometry method, and electrical conductivity, soil salinity, and cation exchange capacity were measured. And finally, microbial DNA was extracted from soil samples, and the purified DNA was stored. In the quantification of bacterial or fungal community size, first bacterial and fungal community size are determined by qPCR analysis, and standard curves were plotted, and average efficiency of bacterial and fungal PCRs were estimated. In the next part of the experiment, a bacterium uh, 60, 16 sRNA gene amplification on sequencing, PCR amplification or hypervariable B regions V3, V4 of this bacteria. Gen was carried out for 45 soil samples, and next PCR applicants were purified with basim and quantified. And finally, applicants were polled at equimolar levels and buried. Sequencing was performed on Illumina Me sequencing platform. In the fungical ATS amplification and sequencing, PCR amplification of fungical ITS1 regions were performed for a total of 45 independent soil samples, and the amplification and sequencing were performed. In the sequence analysis, microbial community bioinformatics was performed using um, QIIME2 2019. Sequences were quality filtered, denoised, and merged. Next, non-unique amplicon sequence variants were alleged with the mapped aligner, and finally data analysis was performed. Okay, this is the results. The major soil physicochemical properties of the RCD and RM farming soils are shown in this table, the table 1. All soils were alkaline with pH value ranging between 8.75 and 9.02. However, a significant difference in soil physicochemical properties were not observed among soil layers in the 12 years RCT model, with the exception of EC. Does the long-term effects of RCT model of soil properties may be more prevalent in deeper layers? Um, Rice productivity traits, including panicle numbers, panicle setting rate, grains per panicle, seed seedings rate, 1,000 grain weight, and gel are shown in this table, the table 2. And panicle seeding rate, 100 grain height, and gels were higher in RCD plots than in RM plots. Panicle numbers, grains per panicle, and seed seedings were not significantly different between RCD and RM grown rice. Okay, in the effects of the RCD model on microbial community composition, diversity, and size, a total of 64 bacterial phyla, two, 2,000 200 sorry 271 classes, 845 44 orders, 1,724 families, 3,140 14 genera, and 5,153 species were detected among of bacterial communities, and the most abundant bacteria phyla were Proteobacterium, Chloroflexin, Acidobacterium, Nitrospirae, Actinobacteria, bacteroidetes, etc. And a total of 15 fungical phyla, um, 52 classes, 199 orders, 310 families, 638 genera, and 1,133 species were identified among the fungical communities. And the most abundant fungical phyla were Ascomiota, Ascomycota, Basidomycota, Morti, Mortierelomycota, Roselomycota, etc. etc. The network structure of the co-occurrence bacteria communities is shown in this figure, the figure number 4. A separate bacterium fungical co-occurrence network indicated a larger proportion of negative correlation in 12 years RCD communities compared to 5-year RCD and RM communities. On the bacteria communities in the 12 years RCT, soils were more interconnected than in the 7 years RCT and RM soils. Notably, fungical network uh, nodes were are categorized as connector. Okay, discussion. Okay, according to the results, and the effect of the RCD model on recycled nutrients and ultimately increase the concentration of nodal nitrogen and available nitrogen. Further, crayfish and turtles can penetrate the surface and base layers of rice paddy soils, thereby increasing soil permeability, community composition, diversity, and size. And according to Falkowski and collaborators in 2008, and Patricia and collaborators in 2010, soil microbial communities in ubiquitous and indispensable ecosystems complement that sustain critical processes like organic carbon turnover, nutrient use efficiency, and productivity. Therefore, microbial community can be used as indicator to effectively describe soil health. And there are city practices incorporating a crayfish and turtle into grouping system can increase the decomposition of uneaten feed, mold, and excreta, thereby promoting nutrient cycling and soil biological health. These processes will consequently enhance soil microbial diversity and microbial community stability compared to the RM soil. And according to Bayer and collaborators in 2010, it's possible that uneaten feed, mold, and excreta of crayfish and turtle can promote fungical dominance in the RCD soils. Therefore, RCD management may promote a greater ability to degrade highly complex organic compounds by soil microbial communities. And according to Sun and collaborators in 2020, rice plants, crayfish, turtles, and microorganisms can use different forms of nitrogen and therefore resulting in highly efficient nutrient utilization. Positive interactions and complementary nutrient use in co-culture systems are the primary foundation for maintaining higher productivity with lesser inputs of chemical fertilizers. Okay. To conclusion, this study demonstrate that the conversion from RM to RCD formed plots could significantly improve soil health by enhancing soil organic carbon, nitrogen, and available nitrogen in the topsoils and deeper soil layers, but also by improving soil microbial diversity. Increased years of RCD farming also increase the modularity in proportion of negative correlations in bacteria and fungal soil community networks, and thus ultimately improving the stability of soil microbial communities. Long term RCD management practices uh, improved soil fertility quality and microbial community diversity, which median increase crop yields. Overall, the study provided a new aspects into the associations of soil health and microbial communities stability with crop yields underlying in sustainability of our city agriculture systems and and the results with further to promote the implementation and application of widespread RCD farming. And this is the reference of the article. And this is all from my part, and thank you very much for your attention.